Hi, this is Miss Litton, and this is 7th period. Excellent students, say hi.
Hi.
And we're getting ready for our first unit exam for the 2019-20 school year. First thing I want to tell you is I am going to print out a PDF of this presentation, so you will have access. You don't have to just take pictures. I'll print it out, and I will post it in Google Classroom in your what? Stream. It'll be in the stream, so it'll be there. And I'm recording this, and knock on wood, the internet has been very glitchy with me, so hopefully it won't quit. And guys, keep an eye on my screen here, because sometimes I won't pay attention, and if it says paused or disconnected, that means it loses connection to the internet, and I need to restart it, okay? So just tell me, that way you have access to it. All right, super smarties, have a calculator at the end. I have a couple games and a practice chi-square problem that's fairly similar to one you might find on your exam, maybe, possibly, okay? Yay, fun, not scary, not scary. All right, so we have 3 topics, right? We have 3 topics: a view of life, chapter 1, basic chemistry in chapter 2, and then the chemistry of organic molecules. Even though the College Board is really only holding you accountable for the organic molecules and the water aspect of chapter 2, I need to hold you accountable for everything, because you're also going to take an SAT 2 at the end of the school year, and you're going to do an awesome job on that, and it's going to expect you to know these things as well. All right, so for chapter 1, first thing we did, let's do this one together. Here we go. Do it together.
Living things are organized, made of cells.
They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. Good. I would probably ask you a question where you would have to say, oh, that's reproducing, that characteristic of life, where you recognize that. So make sure you know what homeostasis means and things like that, okay? We talked about the 4 big ideas. So what's a key word here in big idea 1? The process of what? Evolution. It drives all the diversity, because they're evolving to exploit all these different habitats, but it also explains the unity of life. Because we have a, what would we call that? A common ancestor. And if it's the last universal common ancestor, what is its name?
Luca.
My name is Luca. Good. Big idea 2 is about using what? Energy and molecular building blocks. Are you learning about those molecular building blocks right now? Yes. In order to grow, reproduce, and maintain homeostasis. Third big idea. What is it about? What's the word there? Key word. Pick a word. Information. Okay? How do we store information? How do we transmit that information? We could talk about the genetic code. We could talk about cell signaling, like how we transmit information, and we'll be talking about that. And the last one, I think we would pick 2 words, maybe systems and what? Interactions, right? So those are our 4 big ideas. So moving along, we have life shares common molecules that points to a common ancestry, common processes, similar proteins, and later we're going to have whole chapters just dedicated to the ideas behind evolution, so we'll revisit that. This stuff right here should look familiar, though. What do you see right here? Right here, right. Sparkle pen. There you are. What is that right there? You should know what that is. What is it? Amino acid, right? Do you see the central carbon? Do you see the amino group? What's another name for that? N-terminus. And this is the C-terminus. Good. And this is the H, and then that is the R group. We totally know that. If I was going to add another amino acid onto that, where would I add it? Yes, what kind of terminus would that be? See how smart you are? Oh, wait, let's find another one. Oh, what does that look like? Yeah, a nucleotide, which would be a component of a nucleic acid, right? And I see a nitrogenous base right there. Would that nitrogenous base be a purine or a pyrimidine? Why did you say a pyrimidine? It has one ring. Poor primitive pyrimidines, they only have one ring, right? And we have 3 pyrimidines. If it had 2 rings, it would be called a, and it would have 2. What are the 2 purines? Adenine and guanine, and that spells add, which is stupid, but it helps you. All right? So there's our nitrogenous base. What's this? Sugar. Okay, and it could be one of 2 sugars. What sugar could it be? Ribose or deoxyribose. What would this carbon be right here? Number? Carbon 1, 2, 3. And I'm looking at what prime end if I'm looking here? 3 prime. And it has directionality just like amino acids, yes? 4, chimney makes 5, and this is the? Good. Oh, monosaccharide. What is that? Glucose, a sugar, right? And what are some of the monosaccharides we know? We know we have glucose, galactose, and fructose. Glucose plus fructose is? And that is table sugar. Glucose plus glucose is? And that is maltose. And then, I didn't give you, it's like, I think I said digestion of starch. And then we have glucose plus galactose, and that is milk sugar. Good. Those are disaccharides. Both monosaccharides and disaccharides are used for quick energy. Good. Polysaccharides, we have 2 types. What are the 2 types? Structural and energy. What are 2 structural ones? Chitins and cellulose. Cellulose is for plant, and chitin is found in fungal cell walls and exoskeletons. You're so smart. Structural, we just did that. Energy. What are the 2 energy ones? Glycogen. Who does that? Animals in our liver. We store it. What's another one? And that's by plants. Boom. Look at that. You already know that whole chapter. What is this? That's a fatty acid. What kind of fatty acid is it? Saturated fatty acid. Saturated fatty acids have what? Single bonds and very what kind of chain? Straight. They're easily stacked, and at room temperature they are solid. Whereas unsaturated have double bonds and heat, and those are usually plants, plant fats. Fatty acids, I don't know. Fatty acids, if you have 3 of them, they'll be connected to a what kind of molecule? Glycerol, and we call that a fat. It's one of the 2 types of lipids. What's the other lipid? Steroids. What is the, oh God, there's not video. How would you draw a steroid? 4 rings. What could those be used for? Testosterone and estrogen and progesterone, those are all what? Hormones. And hormones are cell signaling, right? Hormones are cell signaling. And those all came from the precursor of what? Cholesterol. Look at that. That right there, you just did chapter 3 in a nutshell. Are you proud? Power to the people. All right. Look, Luca, last universal common ancestor, right? Similar things in our DNA. Similar, look, I don't know what the click me. Similar sequences. Oh, that's what it went to. We have similar proteins that were coded for the same type of DNA. We have similar errors in many different organisms. We're going to spend more time later talking about that. Last universal common ancestor. Okay. Now, evolution is change over time of organisms, right? And so when the mechanism for evolution, to explain it, there are actually like 4 different things that will contribute to it, one of those being chance. But the one I'm focused on right here is natural what? Selection. What do you hear in natural? Nature. So nature is selecting who is best fit for the environment. When it's selecting those traits, what do we call those? Start with an A. Adaptations, right? Adaptations. Who has the best adaptations to survive in that environment, and you have to what? Reproduce. Good. All right. Big idea number 2, cellular processes. We talked that energy flows and chemicals what? Cycle. Energy flows and chemicals cycle. Remember, do we always, I can't remember because I'm mixing up what I'm teaching honors right now. Did I talk to you about energy, how the laws of thermodynamics, energy is neither created nor destroyed, but it can what? Change forms. But every time you change forms, you lose some as what? Heat. Did we talk about that? Yeah. Okay. And then nutrient cycle. And we'll spend some time talking about that. Let me keep going. Maintain homeostasis. You know that. That's when, do you know that? That's when your environment changes, or maybe your external environment changes, but you try to maintain the environment of that particular cell. Big idea 3, genetics, information, and transfer. We talked about your DNA, and in your DNA you have genes that code for traits. If I'm looking at all of your genes, I'm referring to your genome. We know what DNA is an example of. What is it an example of? Referring to chapter 3. Nucleic acids. What are the other 2 nucleic acids? RNA. Thank you. All right. Behavior. What is behavior? What's the definition of behavior? Yes. Response to stimuli. Okay, so your behavior is a response. Oh, it's right there on the board. Responding to stimuli. Good. Big idea 4, interactions. Oh, we know this. Don't even look at the board. Come over here. We did the first one. Living things are organized, made of cells. Look over it this way. Don't look at that board. And how are they organized? Atoms, molecules, organelles. Wait, do all cells have organelles? No. Who doesn't? Prokaryotic cells are simple, right? Organelles, and then what? Cells. Why do we do this? Where life starts. Cells, tissues, organs, organ systems, organisms, population, same species, community, multiple species, ecosystem brings in the environment, and then biosphere where all life exists. Good. And as you go up in these levels of organization, right, you're going to have these emergent properties, these new properties as you go up. So, right, a circulatory system can do more than just a heart, but an organism consisting of multiple systems can do more than just the circulatory system. Science practices. You are already doing the science practices with your lab. Be familiar with your lab. In case I ask you or reference your lab, what was the independent variable? What's the dependent variable? What evidence did we have? What claims did you make on that lab? What math did you have to use, right? This process of the scientific method. So if you review all those parts of your lab and like your flow chart, you should be good on your scientific method. Okay, we talked about 2 kinds of a hypotheses that we might do. Okay, one is when I look for a direct correlation between 2 things. What is that called? Research hypothesis. I'm saying if I do this, then this will happen, right? But sometimes when you're dealing with data, it is easier not to deal with trying to support a research hypothesis, but failing to support a what? Null hypothesis, which says there is no connection. Right? And then you can use the Chisquare test to analyze your results. And this is your Chisquare test right here. Remember, you take what you observed, right, minus what you expected, square it over your expected. And I have a practice one for us to do at the end, so we'll check that one out there. I do have other practice problems, but I'm afraid we're not going to get through all of them. So I'm going to jump right here to chapter 2. All right. Chapter 2, and if we have time, I'll go back and you can do those other ones, okay? But chapter 2 was our introduction to chemistry. And so let's do the atom story real quick. The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus atomic mass. The electrons are in energy levels. 2, 8, 1832, 8, 8, 8 is great octet rule. If I vary this, what happens to the element? Changes. If I vary this, what happens? I'm making a negative sign. What happens to the element? It's an ion. Gain or loss of electrons, yeah? Why would I ever gain or lose an electron? Stability, right? All right, and then if I vary this, I'm making a circle. What am I varying here? Isotopes, right? I'm varying the neutron number. What we see on the periodic table is an average of all the isotopes. Good. All right. Periods and groups. Remember, periods are on the horizontal, and you're in the same energy level. Yes, like I'm in level 2 or level 3, right? But groups are on the vertical, and groups have the same number of what? Valence electrons. When you know the number of valence electrons, you know how they will what? Interact. So knowing the number of valence electrons determines their behavior, how they will bond or not bond with others. When we talked about bonds, we talked about ionic and covalent. When you share a pair of electrons, that is called a what? Covalent bond. If I share one pair, it's called a single. If I share 2, it's a... Now, if I share pair, if nobody has a stronger pole than the other, that is called a non-polar covalent. Do those tend to be hydrophilic or hydrophobic? Hydrophobic. Because water, which is a polar molecule, because oxygen will share a pair of electrons with hydrogen and another hydrogen, but it shares unfair. So that's called a polar covalent bond. So that means oxygen has it has greater electronegativity. Oxygen has a partial negative charge, whereas hydrogen has a partial positive. So every water molecule will arrange itself as bound water around another molecule that is hydrophilic. And if that hydrophilic molecule is an ion that's negatively charged, are you picturing it? An ion that's negatively charged, what part of the water will turn its face toward it? The hydrogen, because the hydrogen is what? Positive. And that connection, you're not sharing any electrons, that's called a what kind of bond? Hydrogen bond. Do hydrogen bonds only happen with hydrogens? No. And a good example is when we look at DNA, right? It holds the 2 strands of the nucleotides together as a hydrogen bond. Enzymes, enzymes are made out of proteins, most, and when they have their tertiary structure, okay, what can put them in that folding pattern could be a hydrogen bond, but it could also be a ionic bond. It could be a covalent bond, right, that keeps it into that shape. Okay, what's the quaternary structure? Multiple chains. Good job. All right, so we reviewed bonds. Okay, look, there's a hydrogen bond. Okay. Now, a really important one to remember are the properties of water. You want to be able to name the property of water and then give a biological benefit for that property of water. And I reviewed that in one of our essay reviews already. So I'm going to say go to that if you want a little help on that. But you name that property of water and then describe the biological benefit of it. Okay, and we said things like high heat capacity, high heat of vaporization. That allows the planet to be cooled. We talked about adhesion and cohesion and the biological benefit of water moving up trees. So you could review that. So definitely review water. Next, pH. pH is a measure of the amount of what? Hydrogen ions. Okay? If it has a lot of hydrogen ions, the pH will be very low, because it's a negative log scale of that, right? Very low, and it will be referred to as a acid. And if I have a lot of hydrogen ions, then I have very few hydroxide ions. And as I move to a weak acid and then neutral, at neutral, I'll have the same amount of hydrogen ions and hydroxide ions. As I move from a weak base to a base, my numbers are getting even bigger. I have fewer hydrogen ions, more hydroxide ions. Every time I change a number, 4 to 3, pH of 4, now I'm a pH of 3. How many fold difference is that? 10. So if I go to 3 to 2, that's another 10. So 4 to 2 is 10 times 10 is a 100-fold difference, right? More hydrogen ions. Okay, and then we looked at buffers. We just introduced the buffer, but basically we said a buffer holds you to that same what? pH, either by absorbing or producing hydrogen or hydroxide ions in order to keep it there. Okay, that was chapter 2. Again, I have a bunch of practice problems, but I want to make sure we get through everything. Okay, we then moved on to organic molecules. Now, organic molecules by definition contain what in them? Carbon. What's the exception? CO2, that's because it's too simple. That's considered an inorganic molecule. And carbon likes to make how many bonds? 4. And you could have things like long hydrocarbon chains, yes? A bunch of carbons and hydrogen. Then you can spice up your hydrocarbon and add on a what group? Functional group. What is a functional group? Yes?
It's basically where it will be like the same no matter like what carbon structure you sort of around.
Or a group of, it's a molecule, right, that has specific properties, right? What's a good example of a functional group that it's a group of atoms joined together and they have a particular property, right? A carboxyl group, C double bond OH. Give me another functional group. Yeah. Amino, which is what? NH2, right? OH is an alcohol, okay? So those are functional groups. Now, together then, we ended up, and you have this chart, right? You have this chart. There's a PDF of it in the notes where it lists the names and it lists monomers and polymers and their functions. And you just want to be familiar with it. You want to be able to create this chart on your own. I modeled that for you in class, right, when we were reviewing. So you definitely want to be comfortable with that. So let's check real quick, and then we'll do a chi-square test. Let me review with you real quick. You are going to be a monster quiz. Log in as your favorite nickname.
Mr. Refrigerator.
Okay. Go again. You have weird names.
Why are you judging your nicknames?
Because you didn't walk out that door. Don't turn around now. Are you in? Are you in? I still want to do a chi-square test with you. You guys, somebody put, this is the last time I'm going to do this. Somebody put an inappropriate name on it, and I'm going to try not to be angry because I don't want to ruin it for everybody else. Don't do that again. You know exactly who you are. Last time. Okay. Oh, there's 50 of you on here. Okay, start. Oh! Whoa! Whoa! 9 students can't join because the game is full. Sorry. Divide into teams. 54 students are participating. We're going to have 9 teams. Did it tell you what team you're on? All right, are you in? You're competing. I'm going to pause you just for a minute here. We're going to do a chi-square test and just a problem. Just a minute. So hang on, we're trying to... Let's go! I'm going to end the quiz. Who's going to win? Mega Melters. Who is winning? second? Carton crunchers. Who's third? Good job. All right, here we go. Key waivers. I can't. Good job. Where are my mega mouse? Good job, megas. Okay, let's review your answers real quick. All right. A hydrocarbon contains. Did I restart it? No. No, I did. Yeah. Okay. A hydrocarbon contains carbon and hydrogen. True. The hydrolysis reaction brings two monomers together. You said false because hydrolysis is about what? Breaking. Which of the following are energy storage molecules? Starch and glycogen. You can see how many of you got it right the first time. Phospholipids make up cell membranes. What are phospholipids modifications of? A fat. You remove one fatty acid chain and replace it with a phosphate group. All right. Amino acids are the monomers for nucleic acids? No. What are amino acids the monomers for? Proteins. Good. Saturated fats do not have double bonds between the carbons. That is true. The secondary structure of proteins depends upon the R groups of the amino acids charged polar. Is that true or false? False. Right? The secondary structure is just because you have a carboxyl negatively charged and an amino group that is positively charged, right? Okay. Uracil is found in what? RNA only. It should be in the lab under, I think it's labeled like alpha dextro something, but it is it's a big jar. Yeah, in the log cabinets, yeah. Let's keep going. Adenine is found in both. You struggled with that. ATP is the energy storage. Yes. Okay, now, I am going to give you a practice chi-square problem. Now listen to me. You're going to be tempted to do it with another. Hear me. Don't. Okay? Don't watch somebody figure it out. Take a note. Solve it. Use a calculator. Use a phone. When you are completely done, then do a double tap on your neighbor's desk, not loud, so you're like, look, I'm done. Okay? Just quietly do a double tap, and then you can compare answers, and then we'll talk about it. Got it? All right. So here it is. And I'm going to pause this for just a minute, and then remind me to start it again. And you do this at home, okay, children? Okay, so hopefully you had a chance to work that out at home. If not, pause, because I'm going to start talking about it. All right, so the first thing we have is we have some data, right? We have fiddler crabs. They observed 22, guys, I've got to be the one who talks, otherwise it'll distract others. There's just too many people in the room. There are 22 fiddler crabs near the rocks, 4 near the sand, and 7 are by the plants. Do fiddler crabs prefer an area? That's what I'm trying to understand. Now, if I was going to write a research hypothesis, I might say, if fiddler crabs have a choice, then they would like to be near the rocks because there's probably more food there, right? But then I would have to support that with my data, yes? So it might be easier just to write a null hypothesis, which is what? Yeah, fiddler crabs have no preference for an area, right? That would be a null hypothesis. Saying there's no connection between what the environment is and where a fiddler crab goes, right? It's still the same research, the same data, but maybe it will help by disproving a null if I do. I don't know that I am. But by disproving the null, I would be supporting the research hypothesis, the alternative hypothesis, which is they actually do prefer something, right? Okay. Now, the next thing it says is analyze the data. Okay, so how many fiddler crabs do I have total? 22 plus 7 plus 4, what is that? Okay. So if fiddler crabs don't care, then what I would expect is to take those 33 fiddler crabs and divide them into the 3 areas, right? What are my 3 areas that I have? I have rocks, sand, and what else? Plants. Okay, so remember there's observed minus expected. If I have a total of 33 fiddler crabs, I would expect how many by the rocks? I would expect 11 by the rocks. I would expect 11 by the sand, and I would expect 11 by the plants. What did I observe? How many were by the rocks? 22. How many were by the sand? 4. How many were by the plants? 7. Okay, what is 22 minus 11? 11. And what do I do with that information? Square it and put it over the expected, which is 11, plus, what is 4 minus 7? 7? Negative 7, right? Is that right? 7, 8, 9, 10, 11. Yes. Square it over 11, plus 7 minus 11 is negative 4. Square it over 11. So 11 squared over 11, I think, is 11. Right? Okay. So this would be 11. What is, that's 49 divided by 11. What's the math there? 4. Okay. And 4 times 4 is 16. 16 divided by 11 is? What is it? 1.45. Okay, so I add up that, and what does that equal? 16 point what? 9. Okay, 16.9, and that is the chi-squared value. Does it look significant? It does look significant. There's something going on there. So I'm going to go check my chart just in case. How many options did I have? 3. So how many degrees of freedom do I have? 2, right? Because you subtract 1. So the area I'm going to be looking is right here. Okay? Uh-oh. What's my number? So less than 1% of the time would that have happened by chance. So did it happen by chance? Probably not. So I am disproving my null, which would then support my research hypothesis, which would say fiddler crabs do prefer where they live. Right? That's the claim I would make now. Based on this data right here, my claim, fiddler crabs do prefer where they are. Because it's not due to chance, because it's a less than 1% chance that it is due to chance. Okay? Have you got that? All right. I'm going to stop the recording here. And then what you can do is, if you want to revisit any bit of this, I'm going to load it up on YouTube right now, and then it is available for you. Okay? Hear me one more time. Listen to me. Listen to me. Your mammalian brain will work better tonight if you have sleep. Okay? If you have sleep. Focus your energies on your weakest areas. Make sure you have a good meal. Maybe take a walk with a friend and you can quiz each other while you're walking. That's good for your body. And go to bed at a reasonable time. This test does not measure your worth. Right? It is going to measure your efforts that you have put forth. No amount of worrying is going to change anything. Do your best with the time that you have. Okay? Have a good evening. I love you. You're welcome. You're welcome. Make good choices. Don't stay up too late.